Since nerve cells have such longevity and are amitotic, they need to be well taken care of. Therefore, nervous tissue presents with a combination of neurons and specialized cells whose functions are to service the neuron. These cells are called neuroglia. In the central nervous system, the neuroglia include astrocytes, microglia, epidermal cells, and oligodendrocytes. Astrocytes are the most abundant neuroglia. Astrocytes support neurons and anchor them to their nutrient supply. They facilitate the exchange of nutrients between capillaries and neurons. They help position neurons in relationship to other neurons. They also absorb leaking potassium to maintain important membrane potentials as well as recycling released neurotransmitter. Microglia come in contact with neurons and monitor their relative health. These cells are macrophages and are able to attack microorganisms or engulf neuronal debris that may endanger the neuron. Epidermal cells line the central cavities of the brain and spinal cord. They form a permeable barrier between the cerebral spinal fluid that fills those cavities and the tissue fluid bathing the cells of the central nervous system. Their cilia help circulate the cerebral spinal fluid, which contains important nutrients and raw materials. The oligodendrocytes are responsible for the production of myelin. Myelin is a fatty protein that wraps the transmissive portion of the neuron and plays critical roles in insulation and speeding the conduction of the nerve signal. We will talk more about myelin a little later. The peripheral nervous system has two neuroglial cells. Satellite cells surround the cell body of neurons and play much of the same role as astrocytes. Schwann cells wrap around the transmissive portion of the nerve and form the myelin sheath. Though neurons may have varied organization, their structural units are fairly typical. The cell body presents with branching cytoplasmic extensions known as dendrites. The dendrites represent the receptive region of the cell. Some neurons, rather than having dendrites, present with more complex structures that form capsules, corpuscles, or other complex structures. The axon is the transmissive portion of the cell, and the axon terminal represents the secretory region. The axons of many neurons are wrapped with myelin. In the peripheral nervous system, this myelin sheath is made up of several Schwann cells. The plasma membrane wraps around the axon, forming many concentric layers. The tight coil of wrapped membrane is the myelin sheath. The nucleus and most of the cytoplasm of the Schwann cell end up just beneath the outermost part of the plasma membrane. This outermost membrane layer is known as the neurolemma. Schwann cells along the axon do not touch one another, so there are gaps in the sheath. These gaps are known as nodes of Ranvier. There are three major classifications of neurons. Unipolar neurons have a single short process that emerges from the cell body and divides into a proximal and distal branch. Bipolar neurons have two processes, an axon and a dendrite that extends from opposite sides of the cell body. And multipolar neurons have many dendrites extending from its cell body and a single axon. They comprise 99% of all neurons. So let's review how neurons work. As in the muscle cell, the neuron membrane is polarized. The intracellular side of the plasma membrane is more negative as compared to the extracellular side. Neurons interact with one another through the alteration of the membrane potential mediated by neurotransmitters. The terminal branches of one neuron releases neurotransmitter across the synapse between the terminal branch and the dendrite or receptive region of the neuron. In depolarizing events, the binding of neurotransmitter facilitates the opening of sodium gates, causing local depolarization. Multiple depolarizations on multiple dendrites are processed and summated at the region of the cell body known as the axon hillock. When a threshold potential is reached, sodium gates are opened all along the axon that ultimately causes a change in permeability to calcium at the terminal branches, facilitating the release of neurotransmitter that crosses the synapse of the next neuron in the chain, starting the process in that neuron. The neuronal membrane that precedes the synapse is referred to as the presynaptic membrane, and the membrane on the other side of the synapse is referred to as the postsynaptic membrane. The propagation of the action potential along the axon is very fast. However, some neurons propagate the action potential faster than others. There are two major factors that influence nerve conduction velocity. The diameter of the axon significantly influences nerve conduction velocity. The greater the diameter, the faster the nerve conduction velocity. The presence of myelin also greatly increases nerve conduction velocity up to a magnitude of 10 times. Myelin allows for depolarization to only take place at the nodes of Ranvier rather than the entire membrane. It is stated that the action potential jumps from node to node and is referred to as saltatory conduction. Here is an example of how neurons regulate the activity of other neurons. A release of neurotransmitter can lead to either an excitatory postsynaptic potential, or EPSP, a depolarizing event, or an inhibitory postsynaptic potential, or IPSP, a hyperpolarizing event. EPSPs and IPSPs may occur simultaneously on different dendrites, thereby modifying the ultimate response processed at the axon hillock. The pictured graph shows the potential changes associated with the typical depolarization and then repolarization events associated with neuronal activation and return to rest. In an IPSP or hyperpolarizing event, the resting potential is lowered. When depolarization occurs, a greater magnitude of potential change is required to reach threshold. The neuron is thereby effectively inhibited.